Thank you. I have to say, people in the facility are usually very well behaved. So what a tremendous selection of talks. I feel so honored to be part of this group. And thank you so much for all the organization that I know has taken a lot of work. So it's impressive. So I have to say also, it's such a delight to present to a community where I don't have to explain what a lamella is. So this is fun. So we're a lab that we do a lot of EM, but we also do nanomaterials characterization in terms of nanoparticles. But of course, for today, we will be focusing on the different EM capabilities. We have, I think, what probably is really familiar with us audience. We have low KV, we have a higher KV TEM that does cryo. We have our fantastic cryoSIM-FIB and cryoconfocal, which we use for quite a selection of different workflows. So for today, I wanted to just present what I think feels like a tiny little innovative step in terms of a workflow where we start with plunge freezing, we do cryofluorescence for targeting, and then we transfer into our cryoSIM-FIB. So again, the workflow that I don't really have to present to everyone, where we are starting with cryofluorescence on a plunge frozen sample, and then we transfer into our cryoSIM-FIB targeting, and we do either volume imaging and lamella prep for cryo-ET. So we love this workflow. I think many people in this room love this workflow. It was a wonderful collaboration between many wonderful people. Kirk Zimick, who's here in the audience, led the Zeiss team at the time, and then we were collaborating with Wachu's group at Stanford with Gong He and Patrick, and then my team with Peggy and David. It generates this lovely data that I think everyone is quite familiar with looking at. But what we found is that there was a weak spot in this entire workflow that really shows up. I don't know if we have a, doesn't. Anyway, I see now why we're jumping at the screen. So the weak spot in this transfer is going from the cryofluorescence for us into our cryoSIM-FIB. And this transfer is where we would have a lot of ice contamination. We would have damage from breaking grids. We would lose orientation. And so while this is absolutely a possible workflow, we just felt like we could save ourselves a lot of pain and anguish and money that users were paying for this if we could make this a bit more robust. So just for anyone who hasn't seen these specific instruments, I'm just going to very quickly show you what those instruments look like. So this is the cryofluorescence. We're using a Zeiss LSM 880, and we have our Linkum cryo stage is mounted there in the center. And an overview of what the Linkum stage looks like when you pull it off of the microscope. From the top view, on the left-hand side is where the dewar is, and on the right is where your sample is located. You remove the little lid, and we can see down inside where your holder with your grid will be placed. So if we look at what this process actually looks like, so you have a TEM grid that can be stored in this box. Of course, this would all be under liquid nitrogen conditions. And the grid is loaded into the Linkum holder. And then you tighten it in place so your grid is secure. And you come in with a magnetic transfer tool that lifts your holder up and it places it onto the cryo stage. And at this point, you would have your TEM grid is located right here in this location. So all of this is great. You get very little contamination. You do your mapping of the grid. You look at the quality of the ice. sorry, I think I just switched over. Okay, so from there we go into our Leica VCT transfer system. The Leica image is far nicer than it looks in the lab. This is what it is in the lab. We've built an environmental chamber around it to reduce the contamination from ice in the environment. But then if you take that off and you're looking down inside it, this is where your sample is loaded in liquid nitrogen. And then we have a sample carrier, specimen carrier here. And this is where your grid would need to be transferred to. Again, this is just another look at what it looks like on the left with the environmental chamber on the right without. And here is head on. So we have to transfer that frozen grid from our linkum stage. So again, our grid is right up there inside the middle of the linkum carrier. And you have to try not to have too much coffee that morning. You need to work as quickly as possible to transfer that grid into the slots in the Leica carrier. And every second equates to a potential glacier that might land on your precious samples. So you need to work very quickly, but again, you need to work very carefully because you don't want to break your grid as you're going through this transfer process. And you'd also like to not lose your orientation. Not that you can't find it back, but it takes time if you've lost the orientation some. So we wanted to just make this whole process smoother and more robust. And so we connected with the team at NanoSoft, them being two gentlemen, Michael and Michael. It makes it very easy to remember their names. And I have no connection to NanoSoft. I don't get any kickbacks from any of this. I just highlight them because they were so nice to collaborate with. And I think many of us, I think you find somebody who's good to collaborate with and it's just a joy to work with them. They're great communicators. They're very thoughtful about understanding the problem, and they tell you exactly what they're going to do, and then they do exactly what they tell you, which is quite nice. So they worked with us to make some modifications to our cryo holders. So this is the standard holder that it comes as when you purchase it. And so what we wanted to do is remove this central area, and then they fabricated a new piece that goes right in there in the center. And this area right here is the perfect space to slide into place the linkum holder. This is all reversible. So if you ever changed your mind and you didn't want to do it, you could go right back to the original. And so the linkum holder, this is what it starts out looking like. And we needed to remove this little area right here, which you can see has been removed in that image. And this is so you're working at these very shallow fib angles, the holder doesn't get in your way. And so now this holder will slide, or the linkum holder will slide perfectly into the Leica carrier like a little cassette. And so this is what the process looks like. You go in with a magnetic tool, you grab it from the bottom, and then you slide it into the Leica holder. And that's it. You're done. Your grid is transferred. You have very little ice contamination, and you've never touched your grid. So you have maintained your orientation. So this was it made our lives a whole lot easier. And then, of course, the last thing is to make sure, I don't know if I switched. I didn't switch. The last thing, of course, was to ensure that the linkum carrier has been inserted far enough inside your Leica carrier that you can still, it's flush. So your shield, your protective shield can be closed. And so this is where the VCT transfer arm slides into the carrier, and it can close the shield and then whisk your carrier away and pump it down under vacuum, and it is now protected. And that this little innovation has really made our lives so much easier when it comes to conducting this workflow. It's so much more robust. We end up charging our users a lot less because it works pretty consistently. And so we've just been very happy with how this has turned out. And if I had another engineering mechanical issue that I wanted to try and change tools or make modifications, I would probably go straight back to Michael and ask them for their help. This is my team. I have a lovely team. I have an amazing team. And I can't say enough about having great people to work with. And I think that I see that in every person I talk with who also speaks so highly of their team. So on that, I will say thank you.
That's great. Thank you so much for a lovely talk. Any questions from the audience? Sorry, Will, I'm making you do the legwork here.
Hi, forgive my ignorance because I'm outside of the cryo world, but why do commercial companies not make the thing that you've now developed? Does everybody use this Lincoln carrier that would then need that? Or do people use other ways of carrying things across?
Yes, good question. So I think there are different kinds of instruments so that you can have workflows that sort of overall are the same cryoCLEM concept, but you have different cryo stages. Maybe you have a different type of microscope. So this is a solution that is specific to the Lincoln cryo imaging. So you could put that on any fluorescent microscope, but you're working with a Lincoln cryo stage. And then it's specific going into the Leica cryo stage in the EM. And so you could do that perhaps on multiple types of FIBs. It doesn't necessarily have to be a crossbeam. So this is not going to be a solution for everyone by any means. You're going to have the same tools. That I think is true. Absolutely.
Like Michael squared.
Yes.
Any other questions? So I guess maybe I can ask. So obviously lots of work on the hardware there to make that work, and qualitatively it's obviously made your experience a lot better. Were you able to do any quantitative analysis around looking at ice buildup, for example, before and afterwards?
Lord, no. We never went back. So I guess the challenge of the ice contamination in this particular transfer is that it's a little bit of a stealth challenge, because sometimes you would see ice build up on the surface of your grid when you go into the FIB-SEM, and you know immediately, let's just stop here, the grid's trashed. But what you might not know is that a glacier has formed underneath your grid. And so you do your alignments, your correlation target, then you go in and do volume imaging and decide, okay, now we're going to come in from the back to mill the lamella. And only then, far later in your afternoon, do you discover that glacier. And so you might not have a glacier, or you might. So once we had this, there was no point in ever going back at all.
And now you've overcome this bottleneck in your workflow, what's next? So what's now the thing that's stopping the effectiveness of the workflow?
Oh gosh, this is one of those 5 minutes, please. You know, I think one of the biggest concerns that comes back from the researchers who we're collaborating with is they say, okay, great, I want to do statistics. I'd like to look at 30 lamella or 50 lamella, and that's still, we're like, gasp. With the PFIB, I mean, those are some amazing talks I've seen. Maybe you can get to something like that where you're having statistics, but in our situation, it's pretty much a bottleneck of how many lamella per grid. I mean, we can, with correlation, maybe we'll have 6 in a day that look good. So that's still a challenge.
Fantastic, thank you. Okay, if there are any more burning questions from the floor, then we'll just thank Abigail and all of our speakers in today's session.